This is Dr. Stricker. The title of my talk is Lyme Disease, the New Pharma Frontier. I have changed the title of my talk somewhat because over the past year there have been at least 4 novel antiviral drugs for COVID-19 that have been developed. And it's somewhat disappointing that we haven't seen the same kind of drug development for Lyme disease over the years. I'd like to focus on that type of drug development. So Lyme disease has been called the new great imitator. Of course, the original great imitator was syphilis, and both diseases are caused by spirochetes. This is a spirochete that looks like Borrelia burgdorferi, but in fact, this is the spirochete Treponema pallidum that is the agent of syphilis rising up from the culture soup. The Lyme spirochete and syphilis spirochete look very similar. Syphilis has a few more turns, but otherwise they look very much the same. Now, there have been at least 21 different species of Borrelia that have been described around the world. These different species have different pathologies and different distributions, and in fact, not a lot is known about many of these species. And this is an area where Lyme disease deserves a lot more research. There is one particular variant of Borrelia called relapsing fever Borrelia that has become more prominent around the world. And you can see on these maps of the United States, on the left, there's the distribution of Borrelia burgdorferi. On the map on the right, there is the distribution of tick-borne relapsing fever, and you can see there is a lot of overlap of these two species of Borrelia. For Borrelia burgdorferi, as you can see, there is a lot more patchy distribution compared to what we usually have been told about the upper Northeast and upper Midwest and California distribution. And this makes it much more difficult to predict where people will get infected with the Lyme spirochete. Now, Lyme disease is transmitted by ticks, and particularly Ixodes ticks that transmit Borrelia burgdorferi. But for those of you who know about ticks, this is not an Ixodes tick, as you can see from the white dot on its back. This is, in fact, an amblyoma tick. And amblyoma ticks have been expanding their distribution across the country for the last few years. Amblyoma ticks can be found in almost every state now, and this makes, again, it much harder to predict where people will get a tick-borne disease such as rickettsia and ehrlichia, which are transmitted by these ticks. Ticks have been around for a long time. This is a tick from 15 to 20 million years old that was found in the amber fields in the Dominican Republic. It is in fact an amblyoma tick, and it was found to contain monkey blood, and the monkey blood was found to contain a Babesia organism. So ticks have been around for a long time, and there's really no way that we're going to be getting rid of them anytime soon. Ticks have been called sewers of infection. Anything that they feed on, they can pick up and transmit to you. And there are a number of co-infections that go with the Lyme bacteria. And those are shown on this slide: Babesia, Anaplasma, Ehrlichia, Bartonella, Rickettsia, Tularemia, Mycoplasma, and chlamydia-like organisms have all been transmitted by ticks and can cause disease in humans. So the point is that Lyme disease is really a much more complex disease than we used to think because there are these co-infections that often accompany the Lyme bacteria. And this slide just shows the distribution of co-infections associated with Lyme disease. Babesia is usually the most common, but Bartonella is also fairly common. And it's important to recognize these co-infections because the treatment for these infections may be different from the treatment for Lyme disease. Where do ticks bite you? Well, this slide shows the locations. The thigh is actually the most common place, the foot the least common in this study, but basically any place on the body could be a target for ticks. And if you have a rash somewhere, it's important to consider a tick bite no matter where the rash appears. Lyme disease does involve multiple organ systems. The slide shows the different areas of the body that are involved. The immune system, heart, muscles, skin, brain, eyes, nerves, and joints all can be involved with Lyme disease. And again, this makes Lyme disease a very complicated disease and sometimes difficult to diagnose because there are such protean manifestations of the infection. Now, the characteristic hallmark of Lyme disease is the erythema migrans or bullseye rash. Which is a ring-like rash with an expanding ring and a clear center. This, however, is not an EM rash. This is in a patient who said that this was the itchiest rash that he had ever had. And of course, this is not Lyme disease, this is ringworm. And it's important to ask the patient about that. Does the rash itch? And if they say yes, it itches terribly, then it's probably something like ringworm. If it doesn't itch at all and they just found the rash incidentally, then it could well be a Lyme rash. Now, Lyme rashes come in all shapes and sizes, as you can see on this slide. Some of them are polygonal, some of them are look like bruises. There's a big variation in Lyme rash. Only about 9% of Lyme rashes are actually bullseyes, so it's important to remember that. And again, rashes can occur in any part of the body and can look like bruises or rings or solid rashes or have a lot of different shapes and sizes. In medical school, we were taught that the only infection that gives you a rash on your palms and soles was syphilis, but we now know that rickettsial disease can also cause that distribution of rashes. This is a patient who has Rocky Mountain spotted fever with a rash on the palms and soles. So it's important to consider this in the differential diagnosis. Lyme arthritis with a big swollen joint, as shown here, is actually fairly uncommon. Only about 20% of Lyme patients will have frank joint swelling and arthritis. And in fact, this is not Lyme arthritis. This is a swollen joint due to gonorrhea in an exotic dancer. So it's important to consider other diseases as well when you have this type of arthritis. Lyme disease more often causes pain, and the pain can be in the joints and the muscles and different parts of the body. It can migrate. This is a much more common symptom in Lyme disease than a true swollen joint arthritis. Bell's palsy is also a hard sign of Lyme disease, as shown on this slide, with a partial facial palsy. However, Bell's palsy is also quite unusual as a symptom of Lyme disease, and it's much more common when you're looking for Lyme disease to see a patient who looks like. Yes, George W. Bush did have Lyme disease. He got infected either in Camp David, Maryland, or on his ranch in Crawford, Texas. He never talked very much about his Lyme disease, and it's really a shame because he could have been a spokesperson for the disease, but he has never really come out and talked about it very much. Other famous people have gotten Lyme disease. Author Amy Tan, Hall of Fame pitcher Tom Seaver, folk singer Pete Seeger, and rocker Daryl Hall all had Lyme disease. Actor Richard Gere, singer Avril Lavigne, and actor Alec Baldwin, who had some very unfortunate events lately, all had Lyme disease and recovered. Allie Hilfiger, who's the daughter of clothing designer Tommy Hilfiger, had Lyme disease and documented her ordeal with neurologic disease in a book called Bite Me, which is very interesting. It's a first-person account of neurologic Lyme disease, and it's a very instructive piece of writing. And then Yolanda Foster Hadid has become a spokesperson for Lyme disease, has really raised awareness of the disease. And the guy on the right might surprise people. Michael J. Fox has raised millions of dollars for the Parkinson's Disease Foundation. What people often don't know is that Michael J. Fox, before he had Parkinson's disease, had Lyme disease. And he claims that the Lyme disease was treated and went away, and then he developed Parkinson's disease at the age of 30. But you really have to wonder whether the two are somehow related. So in considering Lyme disease, we need to look at three aspects. First of all, define the scope of the problem. Second, develop better testing for Lyme disease. And third, develop better treatment for Lyme disease. And the goal is to implement precision medical care for Lyme disease patients. So first of all, defining the scope of the problem. More than 476 cases of Lyme disease caused by Borrelia burgdorferi are diagnosed each year in the USA. Thus, Lyme disease is 6 times more common than HIV/AIDS, 20 times more common than hepatitis C virus infection, and 30 times more common than tuberculosis in the United States. Ixodes ticks carry more than 237 bacterial species, and new species related to the Lyme spirochetes, such as Borrelia miyamotoi and Borrelia mayoni, are being discovered all the time. Birds play an integral role in the wide dispersal of Borrelia burgdorferi infected ticks. Rodents such as mice, shrews, squirrels, and chipmunks are the primary reservoir for Borrelia burgdorferi, and deer act as quote, mass transit, unquote, and bed and breakfast for infected ticks. Culture of viable Borrelia spirochetes in human genital secretions suggests that Lyme disease could be transmitted by intimate contact from person to person. So again, rodents are the main reservoir for Lyme disease, and lizards can also be hosts for ticks, as shown on this upper right panel. Deer are the mass transit for ticks carrying Lyme disease, and in 1900, there were about 500,000 deer in the United States. By the year 2000, there were 35 to 40 million deer, and that gives you some idea of why the distribution of Borrelia has increased so significantly over the last century. Reptiles can also harbor ticks. This is a python from Australia that's infested with ticks. And alligators also get into the act. You can see this tick sitting on top of this saltwater crocodile. And despite its thick skin, ticks can also feed on these reptiles. Birds, as I mentioned, are a major source of Lyme distribution. These are ticks on a finch feeding merrily away. And birds can distribute ticks far and wide because of their migratory patterns. And then, of course, our best friend, dogs, can also be infested with ticks, as shown in this slide. And dogs can also transmit, carry ticks into the house and facilitate transmission of Lyme disease to humans. So now it has been stated in one of the foremost medical journals, the New York Times, that there is no credible scientific evidence for the persistence of symptomatic Borrelia burgdorferi infection after antibiotic treatment of a tick bite. And that statement has proven to be untrue because there are numerous studies from Europe and the United States showing that there is persistent infection with Borrelia burgdorferi in patients who have persistent symptoms of Lyme disease. So the entity of chronic Lyme disease is a reality. And this is shown by some recent work by John Alcott's group. They looked at a database of 47 million persons enrolled in a wide range of U.S. commercial health insurance plans. 547,000 of those patients were potential Lyme disease cases, and over 52,000 were analyzed. And in those cases, over 63% of the treated Lyme disease cases had at least one diagnosis associated with so-called post-treatment Lyme disease syndrome or chronic Lyme disease. So about 2/3 of Lyme disease patients fail conventional antibiotic therapy and go on to have persistent symptoms. And this slide just shows you the comparison between symptoms in early Lyme disease versus chronic Lyme disease. Very similar distribution with fatigue, joint pain, muscle pain, sleep issues, cognitive problems, neuropathy, depression, heart problems, and headaches. And these are the kind of symptoms that you commonly see in patients with chronic Lyme disease. And in this study that was published a number of years ago, Lorraine Johnson looked at the distribution of symptoms and the effect on health in patients with chronic Lyme disease. As you can see on the right, in the general population, about 16% of patients will claim that they have fair or poor health. In contrast, 72% of patients with chronic Lyme disease, as shown on the left, will have fair or poor health. And this number is worse than all these other diseases, including congestive heart failure, stroke, heart attacks, diabetes, lupus, multiple sclerosis, liver failure, arthritis, hypertension. So Lyme disease is really a very debilitating chronic disease compared to all these other diseases. So one of the problems with chronic Lyme disease is that there hasn't really been a case definition for chronic Lyme disease. However, in 2018, we did develop a case definition that's shown on this slide, and you can look at the reference for the full article. This has helped tremendously in defining chronic Lyme disease, and it does show that this is a real entity. This is something that's not nebulous or unfounded, and these kind of symptoms do make up chronic illness in these patients. So, and just to summarize that last slide, this is from the movie Under Our Skin about Lyme disease. Lyme disease usually doesn't kill you, it just makes you wish you were dead, spoken by one of the Lyme patients in that film. So now, what about how Lyme disease is spread? We know that it can be spread by tick bites. This is a statement from the CDC website from 2015 saying there is no credible scientific evidence that Lyme disease can be spread from person to person through sexual contact. But in fact, there are a number of animal models that suggest that Lyme disease can be spread by sexual contact, at least in certain strains of mice, and also in beagle dogs. And interestingly, also in Ixodes ticks, it looks like Lyme could be spread by tick sex. So that's a very interesting observation. However, there are other animals such as Lewis rats and Syrian hamsters, which are very inbred animal models where Lyme disease is not transmitted sexually. So it is a rather confusing picture. Marianne Middleveen and her group did a study looking for Borrelia in genital cultures from human sexual partners, and they did find evidence of Borrelia infection in both partners, looking at genital cultures. Interestingly, in one couple, the two individuals did not have Borrelia burgdorferi, they had Borrelia hermsii, which is a relapsing fever spirochete. So again, this is circumstantial evidence that Lyme disease can be transmitted by sexual contact, and this is an area that needs further exploration. And in the picture's worth 1,000 words category, this is a semen sample that shows Borrelia spirochetes on the left and a sperm on the right, which again shows that the organism can be present in genital secretions. So you would think this would be a top priority question. Can Lyme disease be transmitted by intimate human contact? There's been a distinct disinterest in looking at this question, and hopefully at some point there will be more groups that will examine this problem. So now, number 2, develop better testing for Lyme disease. The standard 2-tier testing system that's endorsed by the CDC and the FDA involves a screening test with an enzyme immunoassay or immunofluorescence assay, and followed, if that is positive or equivocal, by a Western blot. This has been the standard testing system that's recommended for Lyme disease. But there are a lot of problems with this algorithm. So the CDC FDA-approved commercial two-tier Lyme testing has a sensitivity of only 46%, which is inadequate for a diagnostic test. By comparison, HIV testing has a sensitivity of 99.68%, so it misses almost no cases of HIV disease. The specificity of commercial two-tier testing is about 99%, which means that there are very few false positives. However, a recent study estimated that in non-endemic areas, the specificity may be as low as 20%. So for many patients, commercial two-tier Lyme testing is pretty much worthless. The high cost of commercial test development and haphazard regulation have hampered improvement in Lyme testing, and better testing is already available, but it needs to be accepted by the medical establishment. So what does that testing look like? This is a study from IGeneX laboratory, and I want to say once again that I have no connection at all to IGeneX other than that I use their testing for my patients. So this is comparing the commercial Lyme testing algorithm with the IGeneX algorithm, which differs in two ways. The first is that it uses 2 strains of Borrelia instead of a single strain to do the test. And second, it uses different interpretation criteria with only 2 positive bands needed for a positive test. And what you can see on this slide, if you look in the upper left, the sensitivity of the commercial Western blot, which is what you're supposed to use, is only about 49%, so again, less than 50% diagnostic accuracy. In contrast, if you look at the IGeneX criteria on the lower right, about 97% sensitivity, which is much, much better in terms of establishing a diagnosis of Lyme disease. So this testing exists, you can order it already, but the problem is it's not really acknowledged by the medical establishment. So what about other tests to confirm the diagnosis? Well, we do have these companion diagnostic tests, which are more indirect tests for Lyme disease. And I want to just mention 2 of those. The CD57 natural killer cell subset is a cellular immune marker that's consistent with Lyme disease. It's very useful for monitoring patients who have Lyme disease, and you have a low level usually that improves with Lyme disease treatment. Testing for C4a, which is an inflammatory marker, is also useful in the diagnosis of chronic Lyme disease, and a high level of C4a correlates with symptoms in patients who have chronic disease. Cytokine expression in Lyme disease has become a trendy topic, and this is a study from John Alcott's group, I believe, showing that there are a number of cytokine changes in chronic Lyme disease. However, cytokines are very difficult to measure, and so it's not easy to look at these changes in a systematic manner. And cytokine testing has really not been very useful in tracking chronic Lyme disease. So number 3, development of better treatment. So pleomorphic forms of Borrelia have been studied by Lenora Gilbert's group in Finland. And here you can see there's the spirochete form, the bleb form, the round body form, and then biofilm-like aggregates that are all pleomorphic forms of Borrelia. And these forms have different susceptibilities to antibiotics and can make antibiotic treatment more difficult. Biofilms are basically coatings of slime that cover the spirochetes and again make immune response and antibiotic treatment much more difficult because they protect the spirochetes. And this is a study from Eva Schappe's group at the University of New Haven using the atomic microscope and showing the morphology of these biofilm colonies, which differ according to the species of Borrelia that's being studied. Persister forms have become a very hot topic, and again, these are forms of Borrelia that are not resistant to antibiotics, but they can tolerate the antibiotics. and then survive until the antibiotics are removed, and then they start growing again. And this has been proposed as a very significant method by which Borrelia can survive in a hostile environment. So now we come to designer drugs, and how could we develop designer drugs for Borrelia? Well, there have been a number of designer drugs developed for other diseases, as most of you know. The HIV protease is a target of treatment for HIV disease. The hepatitis C virus protease and helicase also are targets for treatment of hepatitis C. Most recently, SARS-CoV-2 polymerase has been a target for the development of designer drugs for SARS-CoV-2 and COVID-19. And we've seen some very promising drugs, as I mentioned before. The Merck drug is the most advanced so far, but there are others that should be available in 2022. So what about Lyme disease? Well, Borrelia, this is a study from Ying Zhang's group showing that Borrelia has certain genes that are expressed in persister cells, and the one gene that's most interesting is on the lower left here, and it's the gene for the CLIP protease, which is increased 30-fold in persister cells of Borrelia. So this would seem to be a very obvious target for designer drugs. Now, what does the CLIP protease do? Well, CLIP protease is known as a barrel protease. You can see why. It has this barrel shape with a top and a bottom and then a core. And this suggests a certain function for this protease. And basically, CLIP protease is a meat grinder. It takes misfolded proteins, aggregates, unprocessed proteins, and chews them up and spits them out. So it's sort of the garbage disposal of the cell. So blocking this protease would be a good strategy to treat Lyme disease. So far, we have not had any drugs that do this, but hopefully at some point that strategy will be embraced. Another significant protease in Borrelia is the HTRA protease, which is shown here. HTRA protease has multiple functions, and those are shown on this slide. The protease is involved in chemotaxis and motility, in protein processing, and also degradation and insertion in the membrane. So HTRA is a very significant protease, and that was recognized by a number of people, including none other than the government of the United States of America, as represented by the Secretary of the Department of Health and Human Services, Centers for Disease Control and Prevention. And yes, the CDC has a patent on the HTRA protease. What they're going to do with this patent is unclear. We haven't seen any drugs that target this protease yet, but hopefully at some point we will. And then a very interesting family of proteases is the RNA helicase family. This family is interesting because the NS3 protease, shown here on the lower right, is actually the target of most of the wonderful hepatitis C virus drugs that have been developed. So it is a very significant target for drug development. Now, there are other members of this family, and in particular, if you look on the lower left, the DEAH protease was originally described in yeast, but that actually is also the main RNA helicase protease in Borrelia. And this is just shown on this slide. It's in Borrelia, it's called the HRPA RNA helicase. HRPA has been considered a boss gene because it modulates the expression of at least 187 proteins in the Borrelia spirochete. So this is a very significant protease in Borrelia, and again would be a very good target for designer drug development. HRPA has been shown to be involved in RNA processing and is required for mouse infectivity and tick transmission of the Lyme spirochete, as shown by the study from George Chaconis's group. So again, very, very important protease. And the protease menagerie does have a lot of members, and these are actually proteases that are found in plants. As it turns out, plants have very similar proteases to other types of cells. And again, these would be very useful targets for designer drugs for Borrelia burgdorferi. Now, how could we target these proteases? Well, one approach would be to use existing antibiotics and modify them in some way to have extended effects. And this has been tried with vancomycin, where there's an enhanced form that has other properties such as inhibiting cell wall biosynthesis and inducing cell membrane permeability. These drugs turn out to be somewhat toxic and not always very user-friendly, so I'm not sure where this approach has gone. There has been some work with Babesia, another tick-borne infection, as you saw before. And interestingly, Babesia has been the subject of treatment with other types of protease inhibitors. Babesia can cause rather peculiar symptoms such as fever, sweats, hot flashes, chest pain, air hunger, palpitations, and a kind of peculiar type of hip pain. And it is there have been studies done, one using bortezomib, which is a proteasome inhibitor, to treat Babesia, at least in animal models. And here you can see that with an adequate dose, you can inhibit parasite proliferation with bortezomib. And interestingly, bortezomib in this class of proteasome inhibitors has the most activity against Babesia and has very little activity against human cells, and so could be used as a selective inhibitor of Babesia. So this is kind of a model for the type of treatment you would want for Lyme disease if you had similar drugs that inhibited Borrelia. Now, HIV protease inhibitors have been very successful in treating HIV/AIDS, as many of you know. And the group in Boston, led by Attar Chishti, has looked at HIV proteases in their mouse model of Babesia, and what they found was that two of these protease inhibitors were very active against Babesia, as shown on the slide. Lopinavir in red, atazanavir in green were both very effective at inhibiting parasite proliferation. And here again, a study showing that adequate doses of lopinavir and atazanavir can inhibit Babesia, and interestingly, another protease inhibitor, fosemprenavir, was not effective in inhibiting the bacteria. We have actually used lopinavir regimens to treat patients with refractory Babesia, and about 2/3 of those patients do quite well with lopinavir regimens. We're not quite sure why some patients respond to this and some don't, but in patients who've had persistent Babesia infection that's failed 2 or more courses of antibiotics, lopinavir can really be a game changer for this infection in humans. So now, why should pharma develop better drugs for Lyme disease? And here there's kind of the carrot and the stick. So the carrot is that if you look at the annual rate of new cases of HIV/AIDS, it's about 50,000 per year. The total annual revenue of HIV treatment is $18.5 billion. For annual new cases of hepatitis C virus infection, only about 13,000 new cases a year, and the total annual revenue for hepatitis C virus treatment is $4.5 billion. So with annual new cases of Lyme disease at greater than 476,000, what would the total annual revenue of novel Lyme treatment be? It boggles the mind to think of that, but probably it's a lot of money. So that is the carrot. What about the stick? Well, with AIDS drug development, we had all these demonstrations with trashing of pharma booths at meetings, and we had a die-in at the Merck headquarters to make the point that these drug companies should develop better drugs for AIDS. The Lyme community has gotten sort of fed up with the lack of progress with designer drugs, and at some point you may start seeing the same kind of protests with Lyme disease. So in conclusion, the scope of the Lyme disease epidemic is much larger than HIV/AIDS, hepatitis C virus, and tuberculosis in the U.S., and may involve sexual transmission. We need better testing using companion diagnostics for Lyme disease on a national level with support from government agencies. We need better treatment for Lyme disease using designer drugs similar to the ones that have changed the course of HIV/AIDS and hepatitis C virus. And we need Big Pharma to take the lead in the global fight against Lyme disease. And here's my field of dreams. You can see the ticks in there somewhere, but if you build better drugs, the patients will come and will get treated. And if you want more information, please visit the website of LymeDisease.org at www.LymeDisease.org. Thank you very much for your attention.